Hello and welcome to this presentation on ion channels and neuronal communication. My name is Donald Goff. The objectives of this presentation are: explain the role of ATP-driven pumps in creating electrochemical gradients, identify the parts of neurons, examine how voltage-gated ion channels propagate an action potential down the axon, explain the mechanism of acetylcholine release in a chemical synapse, compare and contrast ligand-gated ion channels to voltage-gated ion channels, explain the action caused by the acetylcholine on nicotinic receptors that are type NN and type NM, and examine two different mechanisms of action by two toxins. ATPase, or sodium-potassium pump, is an electrogenic transmembrane ATPase. It's located on the cytosolic side of the outer membrane, and the pump is driven by ATP. This is not going to work with diffusion, but we're going to create an electrochemical gradient by pumping ions against their gradient. So ATP is binding to this pump, and it will phosphorylate the pump, and it will allow 3 sodium ions to connect into these spaces right here, and as it closes, the 3 sodium ions are basically pumped out of the cell, and 2 potassium ions then will be pumped into the cell for every ATP unit that is consumed. Sodium and potassium move against their concentration gradients, which is why it has to be powered to do something to where it's going against the gradient. It plays a critical role in the production of neuronal action potential. So the difference in ion concentration will actually polarize this cell membrane and allow a cell potential or an action potential to be used in neurons. Excitable cells. The ATPase pump action causes the cell to lose more positive than it gains, and it acquires then a net negative charge inside. So since 3 sodium ions are being pumped out of the cell and only 2 positive potassium ions are being pumped into the cell, so every time it pumps there's a net loss of 1 positive to the extracellular space, and that creates a differential that is going to be negative on the inside of the cell. Transmembrane electrical potential is about 60 millivolts. So that's the difference in the ionic charges on both sides of the membrane. Once again, the inside of the cell will be negative, and the outside of the cell will be positive. There are 2 types of calcium ion transport ATPases. One that pumps calcium to the extracellular medium, and then there's another one that pumps calcium from the cytosol to the endoplasmic reticulum inside the cell. And so this will cause the calcium ion concentration to be very low within the cell. So what you'll have is a high concentration of sodium ions and calcium ions that will exist outside of the cell, but there will be a high concentration of potassium ions that will exist inside the cell. Neurons are specialized cells that can carry an action potential. The typical neuron is composed of a cell body. So here's the cell body, And it's also composed, so these are dendrites that are coming off the cell body, an axon, and then down here the presynaptic terminal. Dendrites receive signals from other cells. The cell body is for basic cellular function. So inside the cell body, inside this area right here, you would have the nucleus, you would have mitochondria, all the organelles to perform basic cellular functions. And the axon is this long single projectile that can carry the action potential down to the presynaptic terminal. Voltage-gated ion channels. Voltage-gated ion channels are classified by the ion that they conduct. And so there are voltage-gated ion channels that conduct sodium ion or potassium ion, calcium ion, and some that do chloride ion. But the three that are essential for neuronal signaling are the sodium, potassium, and calcium. And voltage-gated ion channels such as the sodium have voltage-gating sensing domains. So there's basically two different domains on these voltage-gated ion channels. One is a voltage sensing domain, and it surrounds the pore domain, which basically is where the ion will go through once the voltage sensing part of it causes the pore to open. And so depolarization causes the movement of parts of the voltage sensing domain to move towards the extracellular surface. So it causes it to rise up. For instance, in some sodium voltage gates, the S4 will start to move towards the extracellular surface, and what that will do is it will cause the motion to happen, and that motion is transferred to the pore domain by intercellular linkers, which then cause the conformational changes in the pore domain, which then opens the channel and allows ions to flow. And so what we get here is then an action potential. And so voltage-gated ion channels that are sodium and potassium run along the entire length of the axon. And the voltage-gated ion channels are closed until local depolarization happens in response to some type of neurotransmitter. And then what will happen is the voltage-gated sodium ion channels will open for milliseconds. That's because the sodium can flow in at a rate of about 10 million per second. And so the sodium ion flows into the cell and it reverses the polarity. So here's a picture up here. You can see that the action potential causes the sodium ions to come into the cytosol from the extracellular place. And so the inside used to be negative and the outside was positive. Now we have reversed that polarity. In response, though, the voltage-gated potassium ion channels open and potassium flows out of the cell and repolarizes it. So notice that the action potential moves down in the second picture, and so now in response, potassium ion channels open up and repolarize the cell membrane. And so you can see that there's this action potential moving down. These are differentiated by time. And so the potassium ion channels open up split second after the sodium ion channels. And so the voltage-gated sodium ion channels are unable to reopen. If they could, then there could be some sodium leaking back in after the potassium has been pumped out, and that would not allow the a unidirectional wave. But since the sodium ion channels are unable to reopen for a few milliseconds, then that guarantees a unidirectional wave down the axon. And so here we have basically in our action potential, synapse is formed where a presynaptic neuron meets a postsynaptic neuron. So in these pictures right here, this is the presynaptic neuron, and this is the postsynaptic neuron. And there's actually a couple different ways that they can interface when you're talking about nerve cells to nerve cells. And so there can be an electrical synapse, pores, and so what we can have here is in the electrical synapse there's a gap junction. These are not as common as the chemical synapses, but they are in just about every type of nervous or nerve system, and also found in the brain. So they do happen. And what happens is these pores are large enough for the ions just to continue on down through into the next nerve, and so this happens basically through diffusion. A chemical synapse involves a release then of a neurotransmitter, and so we'll talk about neurotransmitters, and this will be the main focus of the rest of this discussion. So how does the action potential cause the release of neurotransmitters such as acetylcholine? Well, the voltage-gated calcium ion channels exist at the distal end of an axon and open when depolarizing waves or when the depolarizing wave arrives. And so on the far end of that axon in the postsynaptic, the presynaptic nerve, there are some calcium ion channels, and when the calcium and what will happen is the calcium ion channels will open, and calcium will enter the cell near the presynaptic terminal and bind to the protein calmodulin. And so over here to the right, this is a picture of calmodulin. It's actually an X-ray of calmodulin, and you can see here that there are these yellow spheres represent calcium ion. And the calcium activated calmodulin activates calcium calmodulin dependent protein kinase 2. And that the acetylcholine is stored in vesicles that are attached to the cytoskeleton and acted by a synapsin. And what will happen here is that the protein kinase causes the release of the vesicle by phosphorylating the synapsin and releasing it from its actin cage. So the next thing that will happen is there is a chaperone system that exists to facilitate the docking of the acetylcholine vesicles to the cell membrane. And so it doesn't just fall, but there's actually a chaperone system in place. RAB proteins attached to the vesicles are basically GATPases, and so these are connected to the actual vesicle, and then RAB proteins interact with other proteins such as PRA1, Rabfilin, and RIM, and they move the vesicle to the proper location where it's eventually going to release the acetylcholine through exocytosis. But as there's a couple different types of proteins that act as a scaffold. Piccolo and Bassoon proteins, they provide a scaffold to guide the docking of the plasma of the vesicle to the plasma membrane. So how does the acetylcholine actually get released then? Well, there is a snare protein system of about 3 different proteins that mediate the exocytosis of acetylcholine. And the synaptobrevin is a snare protein that is attached to the vesicle. So here you can see the synaptobrevin is actually attached to the vesicle, and and then there's 2 other proteins, synaptosomal associated protein 25, which is also known as SNAP25, and syntaxin protein are bound to the membrane. And so these 2 are right here on the membrane. And as the vesicle approaches, the vesicle attaches to the membrane via the synaptobrevin interaction with SNAP25 and syntaxin. And so they form this connection, and that's going to hold it to the membrane. However, fusion of the vesicle to the membrane is caused by activation of synaptotagmin by calcium ion, and there will be a release then. So what will happen is the cell membrane and the vesicle fuse, and basically it opens up, and the acetylcholine then is dumped into the synaptic cleft. So what does the acetylcholine do next? Well, before we can talk about that, we have to talk about ligand-gated ion channels. Ligand-gated ion channels are also, just like the voltage-gated ones, they're membrane proteins that contain a pore, and they help regulate the flow of ions across the membrane. Just like the voltage-gated ion channels, the ion flux is also driven by the electrochemical gradient. In other words, this is not going to be pumped. It's not going to use ATP. It's just going to flow based off of the high concentration of sodium outside the cell will flow into the cell. So wherever the higher concentration is, it will flow into the cell, and that will cause the depolarization. Ligand-gated ion channels are opened, however, by binding of a neurotransmitter such as acetylcholine at an orthosteric site. And so unlike the voltage-gated ion channels, which depended on detecting the difference in voltages outside and inside the cell, this will actually have a ligand that will be complementary in an orthosteric site to cause the gate to open and close. So other neurotransmitters that can interact with ligand-gated ion channels would be serotonin, glutamate, glycine. But the main focus of this presentation will be on acetylcholine. Receptors that bind to acetylcholine are also known as cholinergic receptors. Nicotinic cholinergic receptors are also ligand-gated ion channel receptors. And so they get their name from nicotine. And so over here to the right, you can see that this is nicotine, and you can see acetylcholine. And especially if you look at the top portion here, how they are very similar in structure. So they would bond to the same complementary sites. But nicotinic receptors are named after nicotine, which selectively binds to nicotinic cholinergic receptors. Nicotine does not bind, however, to muscarinic cholinergic receptors, which are more G protein coupled receptors. And nicotinic receptors are cholinergic because they bind to acetylcholine. There are 2 types of nicotinic receptors. There are NN and NM. NN, the second letter, tells where the receptors are located. The NN will be located on the nerves, and the NM will be on skeletal muscle. NN receptors are located on dendrites of all postganglionic, sympathetic, and parasympathetic neurons. So you will find NN receptors on all autonomic nervous system receptors. So the synaptic cleft is a space between two neurons where the presynaptic neuron is in close proximity to a postsynaptic neuron. And so acetylcholine that is released into the synaptic cleft by the presynaptic neuron binds to the NN receptors on the postsynaptic neuron. So the ligand-gated ion channel on the postsynaptic neuron opens and allows sodium and calcium ions, which are in high concentration in the extracellular space, to enter and depolarize the postsynaptic neuron. And what will happen then is the conduction of that electrical signal will continue into the next nerve and the next nerve, and so on. Type NM receptors are located in the neuromuscular junction. So the NM receptors are located within the neuromuscular junction on the sarcolemma of the muscle cell or myocyte. And so the motor neuron releases acetylcholine, which opens the ligand-gated ion channel on the sarcolemma and depolarizes it. The action potential spreads down the T-tubule and causes the sarcoplasmic reticulum, the SR, to release calcium ions. And so basically here's the sarcolemma in this diagram, and that's where the synapse would be, and it would cause the depolarization, which would run down the T-tubule, and then it would cause the sarcoplasmic reticulum to release calcium. And the calcium then would bind to troponin that causes a change in the conformation that allows muscle cells to contract. And so while the troponin's in the way, the muscle cell will not be able to trigger and shorten, and so, or move to the right. And so the troponin once again binds with the calcium, and that's going to allow muscle to contract. So there are a couple toxins. In fact, there are several toxins that involve the nervous system and muscular system. So there are two I'd like to talk about here: curare and botulinum toxin. Curare is a plant-based poison that South American Indians use to coat the tips of blowpipe darts and arrows. So I remember when I was a kid, I watched Indiana Jones, and one of the opening scenes was there was some blow darts that were being shot at Indiana Jones, or were actually in the cave, and they were poisonous. And so I always wondered if those poisons really did exist, but curare actually does exist, and it binds with acetylcholine receptors. So it's antagonistic on the post-junctional face of the motor end plates, striated muscles. And this is going to actually cause the poison to act as a neuromuscular block that causes flaccid paralysis. And so curare is a potent poison, and causes paralysis of muscle. Botulinum toxin, however, it also causes flaccid paralysis, but it does so through a different mechanism. And so the way botulinum toxin works is it actually will combine with protease. It will use protease basically to cleave the SNAP25. and that will prevent the vesicle from being able to connect to the cell membrane, and without the ability to connect to the cell membrane and fuse with it, it will prevent the exocytosis of acetylcholine, which in turn will cause the flaccid paralysis. Ironically, of course, both of these have led to uses in medicine. For instance, curare, the study of curare has led to advances in basically neuromuscular blocks that can be used for intubation, and botulinum toxin A is commonly known as Botox, and has found multiple uses. One of the first uses was actually helping with facial twitches and things like that around the eye. So, thank you for watching my presentation, and here are my resources. So. This is Donald Goff signing out.